In the last instructional video we had built a tree, we'd requested 100 genes, and we had gotten less than 100. So let's scroll down and look at those tree statistics here. So I asked for single copy genes, 100, and it only found 52. I'm going to show you how to increase this, if at all possible, to 100. And it involves using a different tool at Patrick, the Protein Family Sorter. So I've opened up a new tab because I want to keep that guy open. And let's go into Services and find under Protein Tools, Protein Family Sorter. So we click on that. Now we're going to add our genome groups to this. This is a tool that takes all the protein families in Patrick or within your selected group and allows you to see how many of them are shared and to visualize patterns within those protein families. So under genome group, I click the down arrow to find my most recent ones. So here's one with Buchnera. Buchnera are those endosymbionts of aphids named for Paul Buchner, who was the father of endosymbiotic biology. Ricea, I want to add that group, named for Erich Ries, who was a German zoologist who studied lice. Click down. Blockmania, Friedrich Blockmann, the Blockmann bodies that he had discovered back in the 1880s, and ants. And we found out later that those bodies, those big clumps, were big clumps of bacteria. And that's what's earned him having a genus named after him. Move that over. We need to have these in this. It's pretty much similar to the phylogenetic tree box. We need to add them all together. And then Wigglesworthia. Anybody who's listened to this knows what a fan I am of Wigglesworthia, the endosymbionts of tsetse flies. Move them over. And if you have a private genome, I have a private Wigglesworthia. Whoops. And I start to type it, and if it has a little lock in front of it, it indicates a private genome. So I'm going to add that too. Now, because I have 3 different genera here, I need to use the cross genus families. And Patrick, you can use the local families, which are at the genus level, or the cross genus families, which are at the global level. This is the one I want. So let's click search. And it's going to launch the tool. Now, sometimes we have to wait a few minutes. But what we're going to see when this pops up is here on the left will be a dynamic filter that will show us all the genomes in and also the metadata. And here on the right will be a table showing all the protein families. It'll open showing the pangenome for your selection, meaning it'll show all the genes that they share and all the genes that they have uniquely or shared within different subgroups. When the table opens, you'll be able to see the protein family ID in the first column. It will tell you in the second column the number of proteins within that family. Oh, and here we are. In the third column, the number of genomes that have those proteins, the name of the protein family, the minimum amino acid length, the maximum amino acid length, the mean, and the standard deviation. It's a pretty powerful tool, and we have a webinar series on that I would encourage you to watch. So one of the things we can do pretty quickly is with this filter, you can see present in all families in the first column, absent in all families, and either mixed. Like I told you earlier, it's set at the pangenome, so it's showing me everything. But the codon tree pipeline in Patrick looks for all the protein families that have a single, that are shared, Everybody in my selection has to have that protein family. And moreover, the club is even more exclusive because they have to have only a single copy of the gene. If you have 2 copies, it won't use it unless we give it specific instructions. So to see how big the core genome, the one that they all share, is here, I'd have to click here. And suddenly it's showing me that there are 85. So 85, let's go back to this tab. How come it only found 52? Well, because, for example, here we had 76 genomes, 79 proteins. That protein family, not used. It's out of here. So anything with anything like that, it doesn't get used. Let me show you that you can look at the heat map too and be able to see that even more clearly. When I click on the heat map view, and well, because we have ones that they all share, we don't see any of the cells with a black cell indicating 0. But you see any of these with like these mustard color or these tangerine colors, we can't use any of those genomes so that's a huge number of them. So how can we use something like that? I want to use more of those. These are genes that have more than 1 copy. And there's another thing that we could do too. So let me click back on the table. So remember that we already have, actually I want to leave this here because this is a powerful demonstration of what I want to show you. I want to open up a new tab because I'm all about all my tabs all the time. Okay, and I'm going to do the same thing again. It'll be fast because I've already run it, but this will just cement it into your mind of using the tool and moving stuff over, watching me do it. And I know that it's not at all boring to watch someone repeat the same actions again. We all enjoy that very, very much. Wigglesworthia, you go over, we launch the tool. Now also, remember if it's got more than 1 copy of a particular gene in a protein family, it gets eliminated. But what if it has less? So we can also use this to say we want to see, so let's see, there are 76, let me say 75, number of proteins per family to 50. We don't want to go all crazy here. So right now we have 2,612. Let's filter that to 269. And another thing we can do is I want to go from the most proteins to the least. So I can click on that, and I think that got me to least. So click again, and that'll get me to most. And why did I do that? Because it's going to look gorgeous in the heat map. So let's click the heat map view. And then you can see, so these guys could all potentially, there aren't that many, but we could probably add one. Here, let me open this up so you can see it better. So there, like I wouldn't want to add this guy that a lot are missing up, but these guys that just have single ones that are missing, that might not be bad to add. Okay, so we know that there are more protein families we can draw on if we wouldn't be so exclusive. So I'm going to show you how to do that. So let's go into I'm going to go back to my first tab where I have the job. I'm going to click on services, and then I'll click on phylogenetic tree. Here we go again. We're adding them again. And I know, well, I can't imagine that people would get tired of me each time I click on one of these things to say Buchnera, aphids, Ricea, lice, Blockmania, carpenter ants. Well, okay, You might get tired of me saying those things, but there's no one who would get tired of me saying Wigglesworthia tsetse flies. And then my private genome. Whoops, it helps if one can type. Add that to the mix. I'm going to click on the down arrow there to open up the most recent one, save it in Endosymbiont Trees. And I'm going to call it Adjusted Endosymbiont Tree. And now this is a way, if you've listened to me talk about these things before, that I name things. I'm going to ask for 100 genes. This time I'm going to try, watch me while I click over here, to bring in some of these that have a couple, just 1 or 2 deletions. So I'm going to say 10 deletions and 10 duplications. And the duplications would go to this group, where you could see these guys that had duplications in there, and we'll use one of those genes. And okay, this is a good question. Which one? Which one are you going to use? Well, actually, the codon trees algorithm does a blast between the two and finds the one that's closest in homology and uses that. How cool is that? Pretty cool, right? And so I'm going to launch this thing, and I want this to I'm hoping that this will get me to 100 genes. So I submit that. And I get the message that the job has been submitted. But guess what? I already ran it earlier. So it's like Julia Childs baking a cake. Here is the finished cake. So I click on my jobs folder. I can see this one I just did is already running. But here's one I did earlier. So I click on that, and it was 100. You can see I did several iterations. I showed, this is the one we opened up on, 100 genes, 0 duplications, 0 deletions. I did 100 genes, 0 duplications, up to 10 deletions. And then this one was 10 duplications, 0 deletions. So let's look at this, okay? So I highlight the row and I click view. And we're going to go once again to what? My favorite document, the report.html. So highlight that and click the view button. And oh, look at this gorgeous tree. Look at all the Buchnera. And look, this is what's so cool about these endosymbionts. You see the names here? These are the names of the insect hosts that it was found in. They're all clustering together, which is what you would expect. But for the most part, even the genus clusters together. So I'm not even going to try to pronounce this one, but this is Aphis, which is a type of aphid. Cinaria, these are aphids that are on trees like pine trees. Look at them, they're all clustered nice and neat together. This is like my favorite kind of tree. Blockmania, all nestled together, all snug in their clade. Wigglesworthia, they look so beautiful, don't they? And here, the endosymbionts of lice, all nestled together. And what's fun about this is some of these lice are from gorillas, some are from chimpanzees, and some are from humans. Pedicucola is from humans. This guy is from a chimpanzee. These, oh, I said I wasn't going to use, this genome is from a chimpanzee, this genome's from a chimpanzee, and this one's from a gorilla. How fun is that? But this is what I really wanted to show you. 100 genes requested, 100 genes found. So we have the number of amino acids that were used to build the tree, the number of alignments there, the number of nucleotides. So this is a very powerful, beautiful, beautiful tree. So in the next video in this series, I'm going to show you how to take that Newick file and open it in Figtree so that you can get a better picture, because like with the bootstrap values here, overriding the names, that's not publication qualities. We want you to be able to publish this stuff. And then in the last video of the series, we'll talk about how to cite it and put it into your paper, some of the words that you should use. So thanks so much, and I will see you next time.